my name is Ahmet. I'm a PhD student and research assistant at Middle East Technical University, Computer Engineering Department, and the title of my presentation is Investigation of Multitask Deep Neural Networks for Protein Function Prediction. This is the table of contents of my presentation. As we know, proteins are building blocks of cells and they are involved most of the functions within cells, and functional annotation of proteins is a crucial research area for understanding molecular mechanisms of living beings, identification of disease-causing functional changes due to mutations, and discovering novel drugs. An ontology is a set of concepts and categories in a domain that shows their properties and relations. The main ontology for representing protein functions is gene ontology. In gene ontology, each attribute or a function of a protein is represented by the Go term, and these Go terms are structured, each category is structured as a directed acyclic graph, and these Go terms are connected to each other with different relationships. As we go down on the hierarchy, Go hierarchy, the attributes that are represented by the Go terms become more specific. And statistical and machine learning algorithms have been used in for many years in protein function prediction. And in recent years, deep learning algorithms have been applied as a machine learning algorithm in several areas, such as object classification and detection from images, autonomous driving, and speech recognition due to its successful results. And these algorithms can be considered as a collection of artificial neural networks that have multiple hidden layers, namely deep neural networks, and these networks convert low-level features taken from the input layer into more and more complex features in the subsequent layers. And traditionally, learning is performed to predict a single task. In single task learning, the aim is to learn one task at a time. So we divide large problems into small and reasonably independent problems, and these sub-problems are learned separately. And whereas in multitask learning, multiple targets can be protein using a single predictive model. And multitask deep neural networks are specialized neural networks where the aim is to learn a task together with other related tasks. So these algorithms learn tasks with the aim of mutual benefit. And multitask deep neural networks have three main layers, which are input layer and a shared hidden layers, and finally output layer for each task. And the several important features of multitask deep neural networks have been reported in the literature. First, they implicitly build a relationship between multiple targets, and they provide higher level abstractions by building complex features from raw input data. Finally, shared hidden units enhance the prediction results of the targets having less training samples. A multitask deep learning algorithm won the Kaggle drug target prediction challenge, which was sponsored by Merck. Merck is one of the largest drug companies in the world. And these results draw attention to new deep learning applications in drug discovery. And drug discovery and drug target prediction is a computationally similar problem to protein function prediction. In drug target prediction, given a protein, the aim is to predict drug candidates for the target protein, whereas in protein function, given a protein, the aim is to predict functions of proteins. So both problems are multi-level learning problems which can be solved using multitask deep neural networks. Now I'm going to show our method. And in this study, we proposed a multitask deep neural network solution for protein function prediction. We used the hierarchical architecture based on Go levels on Go directed acyclic graph. And the input of the system is subsequence profiles of proteins, and the outputs are Go term predictions. Here I will describe the method that we use for feature extraction from protein sequences. As we know, protein sequences are strings composed of 20 different amino acids, and our representation for protein sequence is inspired from the bag of features model. A Fisher kernel creates a generic mechanism for incorporating generative probability models into discriminative classifiers, such as support vector machines. 4 steps are followed for feature extraction. The first one is the basic feature extraction. Here, we extract fixed length subsequences from protein sequences. Then basic feature vocabulary. Here we cluster the subsequences and created position specific scoring matrices for each cluster and created probabilistic profiles. Then we quantize the features using feature vocabulary. Finally, protein sequences were represented using feature vocabulary as a numerical feature vectors. And this is a proposed architecture. This is a hypothetical Go graph. In this architecture, we first determine the levels of Go terms on Go hierarchy. Then we constructed trained data set for each level, extracted the features, and obtained features were fed to a multitask deep neural network to get a predictive model for each level. And we performed the same procedure for each level separately. So we have a predictive model for each Go level. And this is the multitask deep neural network structure for a level. Each output node represents a Go term in a level, and created models produce a probabilistic score for each trained Go term, and we determined individual thresholds for each Go term to provide reliable prediction. So if a prediction score of a Go term is greater than the predefined thresholds, we consider the corresponding Go term prediction as a candidate positive prediction. We created several multitask deep neural networks for each level with different hyperparameters, and the models we trained the models with dropout technique. The hyperparameters were number of layers, number of neurons at each layer, learning rate, and dropout rate. And we used TensorFlow for training, and computations were distributed on CPU cores. For training dataset creation, we used Go term annotation with manual experimental evidence codes from Swiss-Prot database. We created 6 training datasets based on number of protein associations of Go terms. Here, our aim was to investigate the relation between size of the training datasets and the performance. For example, one of our training datasets comprised of Go terms having at least 50 protein associations. The other is comprised of Go terms having at least 100 protein associations. And we created 6 training datasets in this way. And we calculated the performance of the system using F-score and Mathieu's correlation coefficient. Now I'm going to show the results. We employed 2 evaluation strategies. The first one is the level-specific performance evaluation, and the second one is the overall system performance evaluation based on Go hierarchy. In level-specific performance evaluation, we calculated performances of level-specific models separately. Here, our aim was to determine how successful a model discriminates between trained Go term, and the test sequences in this evaluation includes at least 1 Go term annotation within the trained Go terms. For example, this is the result for cellular component category for Go terms on the first level. Here, x-axis show different training data sets that we created, and y-axis shows F-score performances. And this line corresponds to performance of the first level cellular component Go terms on Go graph for different training data sets. And results of the other Go levels can be seen in this figure. Here, each line represents F-score performance in different levels. Here, it can be clearly seen that the performance increases as the training data set size increases per Go term. We also checked whether the performances of Go terms are related to their levels on Go hierarchy. However, we could not find a relation between Go levels and prediction performance. And the results are similar to this one for biological process and molecular function category. And these are the average performance results for different data sets for level-specific performance evaluation. Here also you can see that the system performance is increasing as the number of annotations increase per Go term. In overall performance evaluation, we fed all our test sequences to the system, and we provide a Go term prediction if prediction scores of the majority of its parents greater than the predefined thresholds. And we calculated Mathieu's correlation coefficient as 0.75, 0.49, and 0.63 for molecular function, biological process, and cellular component categories. The performance result that I have shown so far was the result of the system when the training was performed using only annotations with manual experimental evidence codes. And we observed that the performances of some Go terms were lower, relatively lower, and these were mostly Go terms having less training samples. And here we also investigated the performance change when all annotations were included to the training. Here we had 2 main objectives. Our first objective was to investigate whether the deep learning algorithm that we use can handle the noisy data. And the second, we wanted to observe that how the performances of Go terms were affected when the training was done using using electronic annotation. And we selected 20 Go terms whose number of protein associations were increased highly when we added all types of annotations in training. And we separated these 20 Go terms into two categories as initially low and high performance Go terms based on their performances on experimental data sets. For example, these are the Go terms whose initial performances were lower. Here, the third column shows their performance results when the system was trained using only many experimental evidence codes. And here, the last column represents the results when the training was done using annotation with all evidence codes. As it can be seen here, there is a performance increase for all low-performed Go terms, which are indicated as green lines. And then we applied the same experiment for initially high-performed Go terms. For high-performed Go terms, in about half of the cases, we observed performance decrease. Here, red lines represent Go terms whose performances were decreased. For the others in the middle, we did not observe a high performance increase or decrease. As a conclusion, we investigated the relationship among the performance of the system and the various parameters. And we also investigated whether the level of Go terms on the Go hierarchy is related to their performance. And the performance changed when all annotations were included in training data set. And our observations were the training data set is one of the key factors that affects the performance of our system. And we could not find any correlation between the levels of Go terms on Go hierarchy and their performance. Finally, the performance of initially low-performed Go terms were increased with the addition of electronic annotations, where there is still room for significant improvement. And this is an ongoing study. We are, as a feature work, we are planning to incorporate different protein features and then optimize the parameter. We are going to run our final models on kappa proteins. Finally, we are planning to release these tools as an open access tool for scientific community. And this is our team. This is a collaborative work between me too and UniProt team at EBI. Professor Dr. Volkan Atal is a full-time professor at Me2 Computer Engineering Department. Dr. Rengül Atalay is a full-time professor at Me2 Cancer System Laboratory and Graduate School of Informatics. Dr. Maria Martin is head of UniProt Protein Function Development team at EBI. And Dr. Tunca Doğan, also a researcher at EMBL EBI and faculty member at Me2. Finally, our manuscript is available at archive in this link. You can check our results and you can find detailed explanation about our study. And with this, I want to thank you for all. I'm happy to answer the questions.
So as we know, deep learning methods is very tended to overfitting. So did you do cross-dataset validation?
Yeah, we performed five-fold cross-validation, and we also applied dropout technique with different parameters to avoid overfitting.
Yeah, I know. That's cross-fold validation. I mean, you train your model on one dataset, and you collect dataset from another source, and use your model to test on the other dataset. Do you do this kind of validation?
Yeah, we separated about 10% of the protein annotations independently as a test set, and we did not use them in training at all. And we used that data set as a validation data set and test data set.
Okay, so you didn't answer my question actually. That's cross-fold validation. So all your data set is still come from the same resources, right?
Yes.
So cross-data set validation is that you collect two data sets from different resources, and you train your model on one source and test on the data set from another source.
Yeah, we didn't perform that, but okay. Yeah, it's a good idea to do that.
Yeah,
we'll definitely look into it.
Yeah, that's probably a future network.
Yeah, thank you.
I have another question. So,
how about we let some other people?
Sure, no problem.
Okay, thank you very much for the talk. In your results for the manual confidence course, or a kind of manual annotation of your processes, you show that molecular function has a fairly high performance for your neural network approach. But then biological process and cellular component, not so much. They were fairly low. And the question is, when you add all sources of evidence for geo annotation, do you observe any improvements for the biological process and cellular component?
Yeah, I forgot to mention the result that I have shown for adding electronic annotations was the result of the molecular function Go terms. And we didn't perform this for biological process and cellular component, but we will definitely do this, and we are going to update our archive paper as we have done. And now, for now, I don't know what will happen when we add electronic annotations to the training.
Do you expect any improvements?
For biological process and cellular component, maybe there will be some improvement, but I don't expect very much improvement.
Okay, thanks a lot.
Hi, thanks for the talk. I have a question related to how you extracted features from this sequence.
Yeah.
So the first question is, what was the dimensionality of your vectors? And the second is, like, have you maybe tried using convolutional neural networks because they are more suited for extracting features from sequence?
For the first question, the dimensionality of the features varies about 1,600 to about 2,000 dimension for each, depending on the category. And for the other question, we haven't used convolutional neural networks, but we are looking in the literature, especially for the Go terms having less training samples, and there are studies in the literature that shows that some deep learning algorithms, such as one-shot learning algorithms, can handle the data with less training samples, and we are planning to apply that kind of algorithm to improve our performance and to increase the coverage, of course.
Hi. You train a different network per layer, you said, right?
That's right.
So does it ever happen that when you make your prediction, let's say in layer 3, you say this protein does not have the Go term, but at layer 4, you predict that it has its child?
Yeah,
does it ever happen, and how do you handle it?
Yeah, it happens, as I said, in overall validation. To provide a prediction for a Go term, we check the prediction scores of their parents, and we apply a simple majority voting. If their parents also agrees on the prediction, we provide that as a positive prediction.
Thank you.
This is a bit of a general comment and a question associated to it. I would suggest, instead of saying protein function prediction, Go term prediction. Because if I bring a biologist here, that level of representation of what is function doesn't help. It's like saying a kinase is a kinase. The biology is much, the detail is much more deep there. And maybe we're not there yet to do that, but just to be precise on not rising expectations above what we can do at the moment. Now, my question associated to that is, how would you do with simple other methods like sequence alignments or alternatively, can you show me an example where you could not predict a function even at the Go level with any other existing simple bioinformatic tools?
Can I answer Rafi's first?
Yeah.
I didn't come to bring down the function.
So just in case you didn't know, this is Rafi. He's a great friend. He's also the organizer of a 3D thing, and he has great comments. So just to answer, so Rafi, if I would say that
Is this still preamble destroying me? Go ahead.
I'm giving the game away. Come on. So if I were to say tyrosine, protein tyrosine kinase, would that satisfy a biologist?
No.
No. What would?
Knowing what the substrate is.
No. Okay. So.
Or what is the role of a particular amino acid in regulating the function of that kinase with another.
So that would be done with biological process. You should really come tomorrow.
We can go so far down.
Yeah.
There will always be a level that will not satisfy everyone.
Okay.
But just stopping at Go terms is way too high, and it's okay.
And if
It's the best we can do, it's fine. It's just that we have not to raise the expectations of biologists to the level that then they go and try to use these tools and say, oh, well, this information doesn't help.
Yeah, actually they do, because we actually collaborate with biologists. So we can talk about that
Some
later.
cases it
You
Okay.
can answer this the second part, which is
I forgot the question.
to show, do you have examples where any other standard bioinformatics tool would not have done the job that you can, even to predict go terms?
Yeah, for now the answer is no, but we are going to compare our result with the simple the other tools and something like BLAST or any other any other tools, and we are going to put it on our paper. For now, yeah, no, because we are still doing this project.
Okay, I'll take chair's privilege in asking the last question before our break. Do you have any idea, you showed the support with the IEA terms. Have you, and some predictions went better and some not so much.
That's right.
Did you have any sense of why that happened? What is the identity of the Go terms that were so predicted and so on? Did you drill deeper into that?
Yeah, for the identity of Go terms, I don't have the results for now, but the performance decrease for Go terms is expected because the electronic annotations are noisy data. So if the performance is already good, and the performance increase is expected, so we mainly investigated if we can elevate the performance of the Go terms that have low performance. So it's happening for all the Go terms, and the other ones, yeah, there's a performance decrease. So it's not maybe logical to use this for those Go terms.
Okay, thanks. So we'll take a 10-minute break, after which we have one more talk and our keynote.